What's up, guys? We're going to hit another one here. We're going to kind of go through chapter 4. This is cell structure and function. It's a pretty long chapter, but we'll try to go through it kind of quickly. I got some questions up on it today, and you should be able to find these in the books. I didn't have anything I went off script on. Okay, you should be able to see this now. Okay, I hope y'all are seeing everything. This thing kind of scares me sometimes. I wish y'all were in here so we could make sure we're seeing this. Okay, so chapter 4 is going to go over talking about the difference between prokaryotes and eukaryotes. Talk about the organelles and endomembrane system, and then microtubules and vesicles, and vacuoles, excuse me, and then I just kind of run through the basic parts of what's in the cell. Okay, so let's just kind of go through it kind of quickly. The first questions that I think on the review sheet was the cell theory. Okay, so check a look at this. We've been looking at the microscopic world heavily since the 1700s, but in the early 1830s they started looking at the cell and came up with a unifying theory or concept of the cell theory with Schwann and a couple of others there. And take a look at what they did. This is what they come up with. These are the guys that did it. This is the theory. All organisms are composed of cells, and that cells come only from pre-existing cells because cells are self-reproducing, and that they are the basic unit of structure and function for all organisms on Earth. That's a pretty cool concept. That's the cell theory, and we believe that now. I will say one of the more interesting things in evolution is how did that first cell come about, which would therefore violate the cell theory. So that's kind of one of those fun contradictions in biology. And here's a plant cell. Plant cell leaf there on the left here, and then here is like the trachea lining of the rabbit, pseudostratified columnar ciliate found in the trachea and the windpipe. A little extra histology for you there. Okay, sizes vary. The naked eye can see this. This is the human egg, I believe. They could label those, and we can see down this is what you can see with the eye down to here, and this is the light microscope. And with electron microscope, you can go down to some of the molecules and stuff. They could have labeled those for us. A little error there in the PowerPoint. So cells range in sizes from a few millimeters down to a micrometer. Okay. Now, interesting when they're talking about surface area to volume ratio. And it's a question: why are we 100 trillion cells? Well, take a look at this picture. If you're one big cell, then your surface area is not very big. Okay, in other words, you're 1 4 centimeter cube. Multiply that out, and your surface volume is 1.5 to 1 ratio of surface area to volume. But if you have 8 2 centimeter cells, you have more cell membrane, and you have all of these have a total volume of 64. But look at the surface area: 96 on this one, 192 on this one, 384 surface. So in other words, you have more total surface area. These 64 1 centimeter cubes in cells instead of 8 2 centimeters or 1 4 centimeter. All of those have the same volume. So when you divide that out, look at that surface area to volume is 6 to 1. Okay, and so we tend to see things down this lower end because it gives us more room more surface area to do things on that cell membrane. Okay. Now, quick one here, another second one here. It says list the 3 main types of microscopes that biologists use today. That's in your book. I'm not going to let you read that and look at it, but it talks about light microscopes, which are the ones with lenses, and that's what the guys were doing when they discovered the cell. I think I told you in class that the word cell also implies that the monks lived in little cubicles called cells, and maybe I might go visit my Uncle Billy at Parchment at the prison cell. So small cubicles, right? And we've seen those movies in the prisons, and they have the cell. So that's where the word cell comes from. Light microscope can only go up to about 1,000 power, maybe 1,200. Okay, and the common light microscope resolutions you can see 2 things as separate that are 2.2 millimeters apart, and that's 500 times better than the human eye. Assuming the resolving power of the human eye is 1. Okay, here is the light microscope looking at some it looks like parameciums, I believe, and you can see how that works. And then the transmission electron microscope is going to shoot electrons through the substance we're looking at. And it has an electron receptor, kind of like a photogrid. It catches those electrons and makes an image. This resolution allows us to separate at 0.0002 millimeters, or 500,000 times better than the human eye, versus the light microscope at 500 times. So that is a power of 1,000 times better than the light microscope. And you get an image formed on a fluorescent screen similar to a TV screen. And you can take a picture of that. And then we came out with, and here is a transmission electron microscope of the Euglena, the same Euglena at 12,500 power versus 200 power. See all those internal organelles? That's how we were able to discover all these organelles we're going to talk about in this chapter. And then later on, probably in the 50s or so, 60s, we developed one where the specimen is sprayed with a thin coat of a metal, and then the electrons beam is scanned across the surface of the specimen, like scanned back and forth, and those electrons bounce off, and you have electron detectors to the side that then connect to a three-dimensional image formed on a TV screen, and you get the surface of what you're looking at. So take a look at this. So in this one, you can see the electrons are shot through, and this scanning coil separates them. Then they're picked up, and so it pops off that surface of this substance we're looking at. There's a specimen, and that image gives us the three-dimensional surface of a cell. Now, not as much magnification, but unbelievable outside edge, and this is where we're seeing a lot of stuff going on in research now. Things happening on that outside of the cell. Very fascinating. Resolution is the minimum distance between two objects that you can still see as two objects. Obviously, that gets better as we go up power. We only have light microscopes in Northwest. I think Ole Miss probably has an electron microscope, and I think the scannings are maybe in Jackson. I don't think maybe Mississippi State may have one, but they're very, very expensive. Very, very, very expensive. Okay. All right. Other ones, they're looking at other different types here. You know, I'm going to skip over some of this. Here we go. There's our three, and we have the one there at the left in our labs. Okay. We can do other little tricks with it with the light microscope. We can have a bright field, stained, a differential contrast, phase contrast, and dark field to see different structures inside. So we were able to even see some of these organelles using different types of staining. All these are cheek cells. Pretty cool picture. Okay, and that brings us to the first big question here. Second big question, distinguish between prokaryotes and eukaryotic cells. Very simply put, prokaryote means pre-nucleus, eukaryote means true nucleus. So if you remember, the eukaryotes are those who have nucleuses and all these internal cellular organelles, and those include, you know, the kingdoms, animal, protista, fungi, right? So plants, animals, fungi, and protistas. Plant, animal, fungi, and protistas. There we go. And prokaryotes included the bacterium, which are very common, right? And the archaea, which is a group of prokaryotes that are extremely unique and have bizarre metabolite, or very unusual metabolite. I say bizarre, yeah, I say bizarre. Crazy ways of getting energy, and they can live in harsh environments. And the word archaea means before or primitive, or way before. So it was, the original thought was the archaea came probably before the bacteria did. But both of these are prokaryotes, right? They're in two distinct domains now, because we discovered that these archaea that live in extreme habitats do have chemical structures similar to the, we actually do some things very similar to what they do. And so that led to this rewriting of our taxonomy by creating these three domains, the bacterium, the archaea, and the eukaryote domain, with us being closer kin to the archaea than the archaea are to the other prokaryotic, not containing nucleus, regular bacterium, which are, you know, a lot of them cause diseases, but a lot of them are just out there decomposers in nature, and we use them all the time in manufacturing products and drugs and stuff. We'll talk more about that. Okay, prokaryotes have three shapes. Both of those groups, they range in size of extremely small, 1 to 1.5 micrometers wide, maybe 2 to 6 micrometers long. They can be round circles called coccus, rod shaped, looking like little medicine pills, like a Tylenol shape, bacillus, and then others are spiral shaped spirillums. And some of these it can be flexible, called spirochetes. Okay, now they have a plasma membrane like we do, a phospholipid bilayer, okay, with an outside cell wall made of peptoglycan, and maybe even a glycocalyx layer outside of that. This is all to protect it, right, and resist it from being hurt or moved, so it's looking attached to things. And here's the shapes. You can see them here. Very common. Here's the proteins. Remember our phospholipid membrane? We got a chapter on that coming up. where it is a phospholipid bilayer with a protein sticking through it, right? And we'll come back to that. Here's a typical prokaryote. The fimbria are halide structures to let them grab a hold of the surfaces. They may have a flagellum sometimes for actual movement, but they don't use the fimbria for movement. They have a nucleoid area where their bacterial chromosomes are. A chromosome usually just have one chromosome, and it is in that area, so they don't have a nucleus, and they may have an extra piece of chromosome sometimes. We'll talk about that one later, okay, called a plasmid, but we don't see one in this picture. And you can see the different parts. Now, interesting enough, they do have an organelle that we have. Now, we have DNA, obviously, multiple chromosomes in us, but they also have ribosomes, which are critical organelle to make proteins. So we see that as one of the characteristics that they share with us. Okay, pretty cool. Of course, the cytoplasm, they got that, nucleoid where the chromosome, circular DNA, or the DNA is, their chromosome. Plasmids, there's that other ring of DNA that sometimes they have. Ribosomes, organelles, where proteins are made in their cytoplasm. And we can see a flagellum, fimbria, and a conjugation pila to transfer DNA so that they can have some type of sexual reproduction here in the prokaryotic cells. Eukaryotics means true nucleus. They have membrane around nucleus with the chromosomes in the nucleus, and we have all these organelles we're going to fly through here in just a minute. And these are cellular structures that have a function, and a lot of them are membrane-bound, membranous type structures that do something for the cell. And we'll be looking at those. Those are organelles. So that's the big difference between eukaryotes and prokaryotes. Okay, kind of cool. Now the origin of the organelles is one of much debate here. There's one. Our next question there is, I asked about the evolutionary relationship between the bacterium, archaean, and eukaryote, and we addressed that, and it's going to come back up, and it's for a little more detail in the book if it don't come back in the PowerPoint. But the next one I got is question 5 is, what is the endosymbiosis theory? Is there any proof of it? Well, one of the more interesting theories is that's come along in my lifetime as a teacher is this one right here. For cells, look at this. As they got bigger and bigger, they needed energy source, and maybe they came to engulf a bacterium that was aerobic that made lots of energy. And that became the mitochondria. And then later on, one of our lines maybe picked up a photosynthetic bacterium that did photosynthesis, and that became the chloroplast in the plants, and that was the division of animals and plants. Now, the Euglena has both the chloroplast and of course, well, all plants have mitochondria and chloroplast, but it's an animalistic plant with a protein with the ability to do photosynthesis in it. And so it's kind of an interesting cat we'll look at later. This led to this endosymbiotic theory that is that these organelles came from former organisms that were engulfed by phagocytosis into a larger organism. The fossil record suggests that the first cells were prokaryotes. Biochemical data shows eukaryotes are more closely related to the archaeome than bacterium. The nucleus is believed to have evolved by invagination of the plasma membrane. The invagination process also explains the origin of the endoplasmic reticulum and the Golgi apparatus. Energy organelles, mitochondria and chloroplast may have originated when eukaryote cells engulfed a smaller prokaryote, and that is the endosymbiotic theory on that. Okay, and we'll see that there is the book gives them details, some evidences. There's DNA in both of those organelles, chloroplast and the mitochondria. They have specialized ribosomes in their cytoplasm that are of bacterial origin. And of course, my favorite one is that they're double membrane organelles. And if you take a cell and engulf it, you got the vesicle it's in, that membrane, plus the membrane of the organism you ate. So that's kind of my favorite one because it's a double membrane. Those are the only 2 double membrane organelles that we have. Organelles themselves can be broken down into these intermembrane system organelles, okay, which are going to be like endoplasmic reticulum, Golgi apparatus, rough endoplasmic reticulum, smooth, and vesicles. And then the energy-related organelles, the mitochondria and the chloroplast, okay, which make our energy for us, okay. All right, and here's a massive picture of an animal cell. I'm not even about to go through that, but you can see all these, and we're going to kind of fly through them quickly because I'm been up here. Actually shot one. It didn't require the recorder rooms. I'm a little irritated. We're going to kind of roll through this and see where we're at. And here's a plant cell with those chloroplasts and the mitochondria both, and that big central vacuole, and all these other organelles, and it talks about what they do, et cetera, et cetera. Okay, so we're going to dive into them here, and we're going to work our way into the eukaryotic cell. So we're going to see that that they all are going to have a nucleus and all these other membrane organelles. Okay, here we go. So we'll start with the nucleus. It is also a phospholipid bilayer inside the cell where the DNA is going to be contained. Okay, it has openings in it called nuclear pores. Okay, and it's double membrane there. The chromatin, which is unraveled chromosomes, are found in this nucleoplasm, and when they condense and coil up, they form chromosomes. And we do that when we get ready to do cell division, right? And so those chromosomes coil up and then they're divided. There's a dark spot like a black eye in the nucleus called the nucleolus, and this is a structure where ribosomes are made. The ribosomal RNAs produce ribosome subunits here. They go out in the cytoplasm and snap together and form the ribosome, which is the organelle where proteins are made by reading the DNA from the RNA that's going to be covered in the nucleus, and that RNA carried out to the ribosome. Okay, here is some nice electron microscope pictures. Look at this. You can see the nuclear pores here, holes in it. There's the nucleolus, and then the chromatin is coiled up out here. I have a question that'll probably pop up on another short answer list. What's the difference between chromatin, chromosomes, and genes? And real simple, chromatin is unraveled DNA. When it coils up, it forms the chromosomes that then allows us to divide it into the X's, and we pull the X's apart during cell division. And a gene is a sequence of a chromosome that codes for a protein. So all of our genes code for proteins, and proteins can be enzymes that do all things in the body. So example, I'm ugly because I had some DNA from my mom and dad that made me make proteins, enzymes that made my face ugly. Some of you are beautiful and you got lucky on the role of genetic dice there. Okay, that's kind of cool. Okay, the ribosomes then there's 3 types of it. Those ribosomal RNA subunits go out to the cytoplasm, snap together to form the ribosome, which is the organelle where translation is going to occur, and this is a process of protein synthesis we're going to cover later, but it is 2 steps. Transcription, where you copy the DNA into messenger RNA, which is the copy that leaves the nucleus. Messenger RNA leaves the nucleus, goes to the ribosome, it snaps onto it, and then it reads the secret coding in it and puts the proteins in the correct sequence that, if you remember, we just covered proteins. The sequence of amino acids determines the protein. And so the DNA tells us what sequence to put the proteins in, and then those enzymes are carried out to the cell to do cellular work, or exocytoted out into the blood to cause other things to be done in other parts of the cell. So information from the gene is copied to messenger RNA. I'm reading it here to you, exported to the ribosome, where it then it has secret coding in it that tells us how to put the protein together, and that is called translation, the building of a protein at the ribosome. And this is the central dogma of molecular biology, that DNA is copied into messenger RNA, and that tells us how to build protein sequences, and then those proteins then do all things in the body. And that is the way genes work. Kind of a quick on that one. Okay, here it is. So here's the DNA, opening it up. There's the messenger RNA being copied as the red. It goes out. It actually doesn't stick out what's still in. It goes out. The yellow ribosome snaps onto it, and these transfer RNAs are going to bring proteins in. What they didn't show here, there's a protein being made. Then it gets going to get put in a vesicle and carried out to the that's a free-floating ribosome, maybe carried to the endoplasmic reticulum. Here is rough endoplasmic reticulum, which is famous for transporting the proteins made by the ribosomes that are on its surface. Okay, we'll see electron microscope with that in a minute. So rough endoplasmic reticulum has ribosomes on it, so therefore it transports ribosomes. Smooth endoplasmic reticulum doesn't have ribosomes, and it is where lipids are made. We'll see that in a minute. The endomembrane system then includes the endoplasmic reticulum, the rough and the smooth, the Golgi apparatus, which is the package shipping department of the cell, and vesicles that transport things, little sacs, little vesicles that transport things and store things in the body, like that big central vacuole we saw in the plant to store sugar, which is their product of photosynthesis. Then they eat the sugar to make ATP. Okay, so they make the cake, they bake the cake and make it, and then they eat it. We eat them, or somebody that ate them. So we are dependent on that sun at some point too. Rough endoplasmic reticulum have ribosomes on them where proteins are made, so they transfer proteins and modify them by adding sugars to the proteins and glycoproteins and modifying the protein itself. The smooth endoplasmic reticulum, no ribosomes, and this is where lipids are produced. It also has a huge role in detoxifying drugs in the liver, synthesizing products, and also can store things. Both of these can form transport vesicles that can carry the products onto the Golgi apparatus, which is named after a British researcher named Golgi. So he's so vain, he named it after him when he discovered that organelle. It does look like a stack of pancakes to me. Here it is. And here's an electron microscope. Actually, that's the rhodoplasmic reticulum. All the dots on those are some people say it looks like it's got rhinestones on it, but those dots represent the rough endoplasmic reticulum, and the smooth is over here without any of those dots on it. I've seen better pictures of that. The Golgi, there it is. There's my stack of pancakes with the syrup dripping off down here. See it? So this just gets the proteins and vesicles, or lipids and vesicles from the smooth, and then it does this is the FedEx, or the transportation center of the cell. The cis face has the vesicles come in, and then new transport vesicles on the trans face bring the new products out after they've been modified, and final touches put on them to make them perfect to work. And then they go out and are exocytosed into the bloodstream. Pretty fascinating to me. The Golgi is shipping the FedEx of the cell. Okay, lysosomes are some of these vesicles not found in plants that produced by the Golgi, and they contain the powerful digestive enzymes and acids to break down cellular debris. When you eat bacteria, when white blood cells phagocytize and eat pathogens, they digest them. So this is the lysosome is very simply intracellular digestion. Okay. Lysosomal storage diseases. Defects in lysosomes includes Taussat disease, a very famous genetic disease among Jewish people because it's in the DNA of Jews that they pass down through the Jewish faith and by marrying within themselves, within their religious group. And these children end up dying at 4 or 5 years old, pretty bad one. And we are working on gene therapy to fix that enzyme. It's an enzyme error in that lysosomes. Here is the lysosome. Here looks like it is producing the mitochondria is making. It's got peroxisome that's breaking down this fragment here. So that it's eating that mitochondria. So mitochondria went bad. It's breaking it down. Proteins then are produced and are rough ER, transported. Lipids are made in smooth, carried by this endomembrane system to the Golgi. Just repeating what we talked about. Lysosomes fuse with incoming vesicles and digest macromolecules in there for whatever purpose that they may need to do. Here's showing once again kind of an overview how these products are made. Proteins made here in a rough, lipids in the smooth. Put it all together, bam, bam. Here's a lysosome breaking something down here. And a particle been brought in, so lysosomes can break down intracellular digestion. And here's secretory vesicles carrying things out of the cell. Okay, peroxisomes is another membrane vesicle, but it has enzymes in it, but it is not made it's made differently. Take a look. The lysosome is similar to lysosomes in that it has enzymes. Its lack of peroxisome membrane proteins results in the ADL. It's active in lipid metabolism. It helps break down lipids, but it catalyzes reactions that produce hydrogen peroxide, and in doing so, it makes some free radicals that have to be broken toxic substance. You don't want that around, so, but it breaks it down into water and oxygen by an enzyme called catalase that has in it. Okay, so they're made different, but they have a different function. They help break things down, but they're not at all total. They're not completely intracellular digestion. Okay, they're active in lipid metabolism and breaking down hydrogen peroxide free radicals. So that's pretty cool. Here's peroxisome here, showing it to you in the cell. Vacuoles, just storage units for the cell. No big deal. We don't have a lot of them in us, but the plants have those big ones to put that sugar in. Here it is. There's that central vacuole of a plant cell. And here's a cool question. I've got here on my sheet here, I've got some questions about the difference between smooth and rust and what's the difference between lysosomes and peroxisomes. But here's a cool one. What are granum and crista? And where could we find them and what are they involved in? Well, if you look here, plant cells have the chloroplast and the mitochondria, so they make the sugar, and then they use the mitochondria to make the ATP to do all cell work. So they have both. So the chloroplast is our energy-producing organelle that have a thylakoid membrane in it, and a stack of these thylakoids form a granum. So a granum is a stack of these green stacks in here inside the chloroplast, and the liquid part is called the stroma. So it has these little folds, little green quarters stacked up, and this is where photosynthesis occurs, where we take carbon dioxide and water and make sugar. So the chloroplast makes sugar, and then the mitochondria breaks the sugar down to make ATP. So plants use that sugar for storage and structural material, and then they use it to make their energy. Okay, so granum are these green stacks in the chloroplast for the light reaction of photosynthesis occur, where the sunlight hits. Mitochondria is a double membrane structure. Look at that, one liver cell may have 1,000 of them, so they vary to different cell types. But it has a interfolded membrane inside of it called the crista, where the electron transport chain is located, and then a liquidy part called the matrix. Okay, so here is the matrix, and those folds are the crista. So in here is each part of cellular respiration we're going to cover next week or so, where we make 32 ATPs here. So this is a energy factory. It even looks like a bacterium, right? It's one of the theories of why it looks like why it got that endosymbiotic theory. Look, it looks like a rod-shaped bacterium, and it's the exact same diameter of most rod-shaped bacteria. So, but this is the powerhouse of the cell, and it has that double membrane, the outer membrane and the crista inner membrane. Thus, referring to that question, the last one, or what is a granum or crista, and where they found. So these are the inner folds of the crista of the mitochondria, and the granum are those green folded discs inside the chloroplasts. Super important for energy. We also have cytoskeleton of protein filaments, actin, intermediate filaments, and microtubules that hold the organelles in place and are the railroad lines for these movement of those transportation vesicles in the cell. Actins are thin ones here that form an intracellular matrix of sorts. I think we have a slide of it here, including some actin filaments that can have the movement of ATP and actin myosin, some of those in muscle. Intermediate filaments support the nuclear envelope, cell junctions, and then microtubules are much, look at this, are help control, are the microtubules formed during cell division at these microtubular organization centers to pull the chromosomes apart, and these little molecular motors called dianins here that move in these KINSESIN, or whatever, I'm not sure I'm pronouncing this, has been a new discovery since I went to school, but these are the little proteins that move the vesicles along the filaments. Okay, so the vesicle moves, not the microtubule. So these slide the vessel down like little motors, little conveyor belts, if you want to call it, assembly line. Here's a better picture. There's a piece of analogy, and this is what the actin looks like under a microscope at 850 power. So that shows that cytoskeleton of the cytoplasm. Intermediate filaments, look at that peacock feather, and you can see the cytoskeleton, intermediate filaments at 1,000 power inside of it. And then here is the tubulin dimers, microtubules showing inside the cell of the actin, I believe, in this one here. Actually, that was actin up there. Microtubules inside the cell to get ready to divide from a chameleon. Kind of interesting, but this shows you that they are internal cytoskeleton, all three of those filaments. Centrioles, a weird little structure here, short hollow cylinder composed of 27 microtubules that are going to be involved, and they in pairs. So a pair of these is a centrosome, and these are the tow trucks of the cell that pull the chromosome apart during anaphase of cell division. They also form the structures in the cilia and flagella for movement. And so they look like a compound head or a bean picker, but these are, here they are right here, and this is an electron microscope of it, but these are intracellular comelones or winches. Y'all know what I'm talking about in the front of a truck, the winch with the wire to pull the truck out. But these are cellular winches that pull things around in the cell, specifically the chromosomes at cell division. Okay, pretty wild. And then they have very similar structures in the cilia flagellum, and they look like that combine, right? And they have then this is where we get the movement of the flagellum of the sperm to swim up, and sometimes like the parametrium of cilia, and they use it to move. So these are for intracellular movement. So if you cut a flagellum, it has these. You can see these cross sections, and you can see these basal bodies that look just like the centrioles, okay, and have very similar structure for movement of the whipping motion of the flagellum. Pretty wild. Okay, kind of went through that kind of quick, but anyway, I wanted to just get it, and we'll talk more about that in class. We may ask any questions about that, but that one, I think I even answered more of the questions that I normally have. So anyway, I'll see y'all in class Tuesday, and I'll try to get 5 and 6 up this week. Just a quick overview of that. But y'all can read these, so it's much better if we have an interaction in class where y'all can ask questions. So we may try to Zoom one of these one night this week. All right, y'all have a great evening, and I'll see y'all Tuesday and Thursday in class. Bye.